Marburg virus disease in Rwanda, 2024. September 11th. Summary. In September 2024, Rwanda reported its first outbreak of Marburg virus disease, or MVD, a filoviral hemorrhagic fever with a high case fatality rate. The New England Journal of Medicine article provides a clinical and epidemiological account of the event, describing patient presentations, diagnostic confirmation, and the response mounted by Rwandan authorities with international support. The outbreak highlighted both progress and vulnerability. Rwanda has invested in public health infrastructure, but managing a pathogen of this lethality required immediate external assistance. The report emphasizes early case detection, strict isolation, and contact tracing as the core response measures. Critically, the article notes the absence of approved antivirals or vaccines for MVD. Management therefore relied on supportive care, fluid resuscitation, hemodynamic monitoring, and treatment of hemorrhagic and multi-organ complications. BBIU structured opinion, monoclonal antibodies beyond cancer, strategic implications for Marburg virus outbreaks. 1. Context: a virus at the edge of lethality. The Marburg virus, a close relative of Ebola within the Filoviridae family, remains one of the most lethal pathogens known to medicine. Its mortality has ranged from 25% to as high as 80% in outbreaks across Africa. Transmission occurs primarily through direct contact with body fluids of infected individuals, contaminated surfaces, or during funerary practices. Unlike COVID-19, it is not a respiratory virus in the classical sense. However, saliva, sweat, and vomit from patients in advanced stages may contain sufficient viral load to generate infectious microdroplets at close range. Pathophysiologically, Marburg targets endothelial cells, hepatocytes, and immune cells. This creates a deadly triad: endothelial damage leading to hemorrhage and capillary leak, liver necrosis with loss of coagulation factor synthesis, and immune dysregulation with blocked antigen presentation, T-cell apoptosis, and a cytokine storm. The paradoxical result is both hemorrhage and microthrombosis, known as disseminated intravascular coagulation. The viral life cycle is rapid. Once inside the cell, replication and budding can occur within 8 to 12 hours. In humans, incubation lasts 2 to 21 days. Clinical progression moves from nonspecific fever to gastrointestinal and liver involvement, and finally to catastrophic hemorrhage, with most deaths occurring between days 8 and 16 of symptoms. 2. The window of intervention. Despite its brutality, Marburg offers a therapeutic window. The incubation plus early symptomatic phases, approximately 10 to 30 days post-infection, present a critical period where antivirals, immune-based therapies, or intensive supportive care could still alter outcomes. Once patients reach the hemorrhagic phase, mortality escalates and therapeutic rescue becomes nearly impossible. This opens a central question. Should therapies considered too risky in oncology, such as immune checkpoint inhibitors or NK cell activators, be repurposed for viral hemorrhagic fevers? The answer lies in recalibrating risk-benefit thresholds. In diseases with 80% baseline mortality, even modest gains justify aggressive experimentation. 3. Monoclonal antibodies beyond oncology. Monoclonal antibodies, or mAbs, have become synonymous with oncology, where they unleash immune responses against tumors by blocking inhibitory pathways. Yet their function is not cancer-specific. They are platforms that can be redirected against viral immune evasion. Checkpoint inhibitors such as anti-PD-1, anti-PD-L1, and anti-CTLA-4 could reverse T-cell exhaustion induced by Marburg infection. NK cell activators like anti-KIR, anti-NKG2A, and bispecific NK engagers could restore the innate cytotoxic response, clearing infected cells before viremia peaks. Bispecific antibodies could theoretically be designed to tether immune effector cells directly to Marburg-infected targets, mirroring advances in oncology. These strategies mirror the precedent set by Ebola, where monoclonal cocktails like inmazibinibanga reduced mortality from 70% to about 30 to 40%. 4. The interferon gamma dimension. Interferon gamma, or IFN gamma, is the master cytokine of antiviral defense. It upregulates antigen presentation, activates macrophages, and enhances T and NK cytotoxicity. Combining mAbs with IFN gamma could amplify immune recovery in Marburg patients. But timing is critical. In early infection, IFN gamma could synergize with mAbs to restore control of viremia. In late stages, where cytokine storms already devastate the patient, IFN gamma may worsen endothelial leakage and accelerate multi-organ failure. Thus, therapeutic benefit hinges on early diagnosis and administration, a challenge in remote African outbreaks. 5. Risk-benefit recalibration. In oncology, severe immune-related toxicities are considered unacceptable beyond a narrow threshold. In Marburg, the calculus changes. With mortality reaching 80%, the medical and ethical question is inverted. Is it more ethical to risk immune hyperactivation or to withhold a potentially life-saving intervention? The Ebola experience proved that experimental mAbs could be deployed under compassionate and emergency use frameworks. For Marburg, the same logic applies. Even if immune activation causes complications in some patients, the overall survival benefit may outweigh the risks. 6. Economic and strategic dimensions. Monoclonal antibodies are notoriously expensive, largely because oncology tolerates high pricing and volumes remain limited. Expanding their use into high-incidence infectious diseases like COVID, RSV, Marburg, and Ebola creates the possibility of economies of scale. Larger production runs reduce cost per dose, broaden global access, and reframe monoclonals from elite cancer drugs into pandemic countermeasures. This has direct geopolitical implications. Countries with stockpiles of antiviral monoclonals would hold strategic advantages during outbreaks. Institutions like CEPI, Gavi, and BARDA could subsidize deployment, while biomanufacturers would be incentivized to expand capacity. The result is both increased volume of sales and reduced production cost, transforming the economics of biologics. Number 7. Structural implications for biodefense. Marburg illustrates the fragility of the current biodefense architecture. If monoclonals can be repurposed as emergency antivirals, they shift from niche oncological products to dual-use countermeasures. But this raises additional concerns: availability, equity, and the potential misuse of filoviruses in bioterror scenarios if countermeasures become reliable. Nevertheless, from a strategic standpoint, monoclonal antibodies represent one of the few immediately repurposable tools capable of altering the natural course of Marburg virus disease. 8. BBIU position. From the perspective of the Biopharma Business Intelligence Unit, or BBIU, the case of Marburg highlights a broader truth. Medical tools cannot remain siloed. Antibodies designed for cancer can and must be redeployed against pathogens with catastrophic lethality. In a disease with near-certain mortality in advanced stages, the ethical, clinical, and economic calculus changes. What once was considered too dangerous in oncology becomes a justifiable gamble in viral hemorrhagic fevers. The potential to cut mortality by even 20 to 30% represents not just clinical progress, but a structural shift in how immunotherapy is conceived. Not as a luxury product of oncology, but as a frontier arsenal in global biodefense. Marburg virus forces us to rethink antibodies, not as drugs of privilege, but as tools of survival. Annex: regulatory, ethical, and strategic considerations for monoclonal antibody deployment against Marburg virus. 1. Regulation and bioethics, patient autonomy in the context of extreme mortality. When facing a disease with case fatality rates of 50 to 80%, the ethical foundation must shift. Patients should not be denied access to experimental therapeutics simply because they fall outside approved indications. Instead, the regulatory framework must ensure transparent informed consent, where the patient or family is clearly told. Baseline mortality is extremely high, the treatment is experimental, with uncertain efficacy and possible severe side effects, and the patient has the full right to decline without prejudice. This restores agency to the patient and aligns with the bioethical principle of respecting autonomy under catastrophic conditions. Number 2. Logistical feasibility. Cold chain precedent in mRNA vaccines. The argument that monoclonals cannot be deployed in remote settings due to cold chain requirements is weakened by precedent. During COVID-19, both Pfizer and Moderna's mRNA vaccines, which required ultra-cold storage, were distributed globally, including in low-resource settings. If such infrastructure was built under pandemic urgency, it can be repurposed for Marburg countermeasures. The true bottleneck is not technical feasibility, but political will, funding, and prioritization. 3. Economics and market expansion, beyond oncology. Pharmaceutical companies typically justify the high cost of monoclonal antibodies by their narrow oncology markets and limited patent windows. However, expanding indications to viral hemorrhagic fevers creates new global demand. Emergency deployment programs such as Gavi, CP, WHO, and BARDA can subsidize purchases. Each new indication may also enable patent term extensions or data exclusivity, increasing return on investment. Thus, Marburg is not only a public health emergency, but also a strategic market expansion opportunity, reframing monoclonals as multi-disease assets rather than oncology monopolies. 4. Trial design, pragmatic Phase 2 protocols. Given the impossibility of large Phase 3 trials in rare outbreaks, adaptive Phase 2 protocols are more realistic. The design should include parallel arms, monoclonal antibodies alone, interferon gamma alone, and combinations. Segmentation of subjects should be stratified by weight, age, sex, liver function profile, coagulation status, and inflammatory biomarkers such as CRP levels. Primary endpoints should include survival at 28 days, viral load reduction, and immune activation markers. Secondary endpoints should include coagulation recovery, liver function, and cytokine profiles. Such a protocol balances urgency with scientific rigor while maximizing learning from each outbreak. 5. The vaccine uncertainty factor. Vaccines for filoviruses remain elusive. Both Marburg and Ebola display immune evasion strategies that can render vaccines less effective or even dangerous by inducing non-neutralizing antibodies that worsen outcomes through antibody-dependent enhancement. Moreover, viral diversity raises the risk of immune escape. This reinforces the need for therapeutic countermeasures, monoclonal antibodies, and cytokine modulators, rather than relying exclusively on prophylactic vaccines. BBIU closing statement. The integration of monoclonal antibodies into the Marburg response is not merely a scientific proposal. It is a regulatory, ethical, and economic imperative. The precedent of COVID-19 proves that cold chain barriers can be overcome. The precedent of Ebola proves that experimental monoclonal antibodies can be deployed successfully under emergency frameworks. And the precedent of oncology proves that monoclonal antibodies can be repurposed as immune recalibrators. In a disease where mortality approaches certainty, refusing experimental therapy is itself an ethical failure.
Welcome to the Biopharma Business Intelligence Unit, where symbolic integrity meets strategic clarity. At BBIU, we go beyond the headlines to decode the structural forces behind global biopharma, AI, and economic realignment. If you value precision, verification, and symbolic depth, this channel is for you. Subscribe now to access our weekly briefings, dossiers, and intelligence reports, and consider joining our institutional tier to unlock early warning systems, custom metrics, and exclusive strategic insight. You're not just listening, you're entering the future of epistemic intelligence. www.biopharmabusinessintelligenceunit.com